Welcome back, microbiologists. This is our final week of experimentation in which we are going to carry out completed tests and confirmed tests for the presence of E. coli in water samples that led to gastroenteritis in the residents at the town of Beaufort on the Genesee River after the Great Flood this fall. So let's get back to water analysis testing. You'll remember from last week that if we're looking for fecal coliforms in water sample, we typically go through a three-part testing procedure. There's a presumptive test, which is just a general screen that helps us narrow down a multitude of water samples to find those that are most likely to have fecal coliforms. You did the presumptive testing last week, and then the very last question that you had in lab notebook number 2 asked you to figure out which of these water samples is most likely to have been the culprit. Which one of these was most likely to be contaminated? What you actually did was you carried out the confirmation test from the information that you had from last week's lab by narrowing down your 4 water samples to the one that has a very good chance that E. coli was present. E. coli, of course, being our indicator species to let us know that there's likely fecal contamination in the water sample. And really what that means is that a confirmation test is just a logical interpretation of a couple different tests that you did last week. From that, then, we can move on to the completed tests, the proverbial nail in the coffin, the tests that say that there is definitely E. coli present here. And not only is it here, but I know exactly how much. And that is what we're going to complete this week. We will finish up the confirmation tests and we'll move on to the completed tests. So one of our presumptive tests was plating water samples on differential and selective media. And we used EMB. We know that EMB contains a couple dyes, eosin and methylene blue namely, that cause bacteria not only to look differently, but to grow on the plate if they are gram-negative. If green colonies grew on our plate from a water sample, then we can presume, but we can't be absolutely certain that E. coli is present. E. coli has a characteristic green sheen on an EMB plate, but could there possibly be other bacteria that also have that same phenotype? Short answer, yes, there actually are. E. coli again is the green one. The second presumptive test you did was to grow bacteria in phenol red lactose broth. Phenol red lactose broth contains lactose as a fermentable carbon source, and when lactose is fermented, it produces an acid that causes the phenol red pH indicator to turn color from red to yellow. Some bacteria, like E. coli, also produce gas during the fermentation process, and if that's the case, they form a bubble inside the tube, as we can see here in the second tube from the left. That gives us a good indication that E. coli could be present, because E. coli will turn yellow and make a bubble. But I don't think that we can be positive that E. coli is present, because there could be other bacteria that also lead to the same phenotype in phenol red lactose broth tubes. Again, spoiler alert, there are other bacteria. But what if we were to do both of these tests side by side? Over on the left hand of the screen, we've got a water sample that we have used to inoculate phenol red lactose broth tube, and maybe one of those tubes turned yellow and had a bubble, which give us some evidence that E. coli could be present, but we're not absolutely sure, because there could be other bacteria that might also do the same thing. Also recall that these water samples are being taken from the environment, so not only are they going to contain one type of indicator species that are found in mammalian feces, but they're also going to contain bacteria that were also present in that environment. Do the same thing on the EMB plate. Some may or may not turn green, which would give us evidence that E. coli could be present, but doesn't rule out the possibility that other bacteria are also there. Both the phenol red lactose broth tube and the EMB tests gave us the presumption that one of these samples could contain E. coli. We're going to move now from the presumptive test to the completed test, in which we're going to take the results of those 2 independent biochemical tests and use them together to eliminate potential false positives, right? Because we don't want to be testing water samples that don't have E. coli, because it's timely and it can be very costly. So as an example, a positive result in the phenol red lactose tube, which was the yellow color, acid fermentation, and the production of gas, is indicative of E. coli. But it is also indicative of Staphylococcus aureus, which is a gram-positive non-coliform bacteria, which also produces acid and gas in the presence of lactose. We can see another false positive on the EMB plates. Another species of bacteria, or another genre, Citrobacter, can form green colonies on EMB, but it is not a fecal coliform. And both of these have the potential to be found in the environment, which would lead us to false answers if we were simply to look at one of these tests at the same time. By moving from the presumptive test to the completed test, we're using independent tests to eliminate those false positives and give us more support for the presence of E. coli if both of those tests are positive. And that's a key point, because Staph aureus will turn yellow and make a bubble in phenol red lactose broth, but it won't do the same thing on EMB. And likewise, Citrobacter will give a positive result on EMB, but not in the phenol red lactose broth tubes. E. coli will be positive for both. So this is what our flowchart now turns into when we take the evidence from our presumptive tests and we make a conclusion called a confirmed test. This was pretty much the same thing that you did in the last question on notebook number 2. You had looked at the evidence from the 4 water samples in your phenol red lactose broth tubes and saw that only one of them turned yellow with a bubble. That's pretty good evidence that E. coli could be present in that water sample, but it's not incontrovertible. Now, when you looked at your EMB plates in the notebook number 2, you had 2 different water samples that produced green colonies. That might have led to some confusion. You say, well, I can't tell which of my 2 locations have E. coli present because there's 2 of them that are green. If you were to look at those results side by side, you would see that one sector of your plate had green colonies, but not yellow with a bubble phenol red lactose broth tube. So that one can't be a positive because it doesn't meet the requirements for our confirmed tests. There's a lot of room for interpretation in these tests. Remember that we do have some coliform bacteria that don't turn green that could be present in a fecal sample that are only pink. Well, that means that if we had a yellow bubbled tube in the lactose broth tubes and something that grew on EMB and appeared to ferment lactose, we could still be looking at a coliform. So it's not hard and true that only green colonies are E. coli. In our example on this slide, we could have 2 locations that would have passed the confirmed test. That leaves us with another question. How do we know which of those is correct, or are both of them correct? With the confirmed tests done, we have a pretty good idea of which water source is our culprit, the source of E. coli contamination. But we need to move a little bit further. We need to get to the completed tests that put together all of the individual bits of evidence so that we have incontrovertible support for our determination. First of all, are we certain that the bacterium that we found in our water sample that was positive for phenol red lactose broth fermentation of lactose, that was green on EMB, is in fact a gram-negative rod? And the way we figure that out is with a gram stain, technique that you used last week. We also need to know how much is present in the water, because that's often very important when looking at the level of contamination. And we do this by enumeration, or counting the bacteria, or establishing the concentration of bacteria in colony-forming units per milliliter, like we've been doing for the past couple weeks. We also need to reconfirm that lactose fermentation. We would call this a recapitulation experiment where we reconfirm what we just found. And we do that in another tube of phenol red lactose broth. And we can also reconfirm that we have green growth on EMB. So let's go backwards and talk a little bit about enumeration, because we have a couple different ways of doing this that are going to be useful for your final project. Enumeration is counting bacteria. We want to know how much is present in the water samples to determine if it's at a dangerous level. We did a bacterial count one way, using plate counting, using serial dilutions to go from a very concentrated culture of bacteria to a much more dilute culture that would allow us to count individual colonies on a petri dish. We're going to do that again because that gives us a very precise, accurate, and repeatable number that's good to know. We can also use some commercial counters, which is something new that we are introducing this week in lab. They are quick, they are only semi-quantitative, so they're not particularly accurate, but they are certainly better than nothing. And we'll combine the 2 of these this week in lab. So let's go backwards and look at dilution plating again. And some of this should be review, and hopefully now with a little bit more context, it'll make a little bit more sense to you. We needed to use serial dilutions to reduce the concentration of bacteria in a sample. because in all likelihood, there's going to be so much bacteria that simply putting a water sample on a petri dish is going to lead us to a lawn of bacteria, too many colonies to be able to count. We also know that bacteria in water will only become visible to the naked eye when it gets to be pretty dense, meaning there's greater than 1 times 10 to the 6th colony forming units per milliliter. I can guarantee you that the water samples that we've been using in lab are crystal clear. I can't see anything in them. So right there, that gives us the indication that however much bacteria is present in our water samples, it is less than 1 times 10 to the 6th CFU per mil. That gives us a good starting point because if we wanted to do serial dilutions, we can just do a couple 1 to 10 dilutions every time, reducing the concentration tenfold until we have a low enough bacterial count that we could be able to count them on a petri dish if we were to plate them. So now let's say that I start with a water sample, and the water sample looks crystal clear. I can't see any bacteria, so I know that there's less than 1 times 10 to the 6th. Let's just cut that concentration in half so that we have a very high density of bacteria that would certainly be considered unsafe by the EPA. That's also far too many to be able to count. If I was to put 1 milliliter of this onto a petri dish, I would end up with a lawn of too many bacterial colonies all growing together. So I can do a serial dilution by taking 1 milliliter of this stock, or our harvested water sample, and putting it in a tube with 9 milliliters of distilled water, thus doing a 1 to 10 dilution so that my concentration of bacteria is tenfold less than we started with, meaning that every milliliter of water in this test tube contains 5 times 10 to the 4th, or 50,000 colony-forming units. Now remember that when we're doing this in practical terms, we can't plate a whole milliliter onto a petri dish because it would just form a puddle. So in practice, we only pipette 100 microliters, which is 1/10 of a milliliter, meaning that if we have 5 times 10 to the 4th bacteria per milliliter, 1/10 of that would be 5 times 10 to the 3rd growing on our petri dish. 5 times 10 to the 3rd is still far too many colonies to count. But at this point, we don't know how many were in our starting tube. At this point now, this is an unknown concentration of bacteria, so we would do 3 serial dilutions until we get down to the point where we're pretty certain we have few enough colonies that would be countable. So carrying on the serial dilution from tube number 1 into tube number 2, do another 1 to 10 dilution. Our cumulative dilution factor is now 1/100th, because this is 100 times less concentrated than we started with. I can do another 1 to 10 dilution by plating 100 microliters onto a petri dish, and if we did that, we would expect to have 5 times 10 to the 2nd growing on our plate. 500 colonies is still quite a few to count. It's reasonable, but doing another dilution, we end up down with 5 times 10 to the 1st, or 50 colonies on a plate, which is perfect. 50 colonies times 10, that gets us back to our tube here, 5 times 10 to the 2nd. We can just continue going backwards with a 1 to 10 dilution, second 1 to 10 dilution, third 1 to 10 dilution. 50 colonies counted on this plate right here means that we had started with 5 times 10 to the 5th. This is the same procedure that you can use by counting colonies on a TSA plate. It's pretty clear that serial dilutions are very labor intensive, and they use a lot of media, and there's lots of places to make mistakes, pipetting errors, mathematical errors. Another way of counting colonies is to use a commercial product, something called a paddle tester. And we have a couple of these in lab that we'll use to do a somewhat semi-quantitative bacterial count. So these paddle testers have 2 different sides. And what we're looking at here, highlighting with my laser pointer, is a plastic vial that's sterile that's containing a plastic paddle that has 2 sides. And each side has some bacteriological media that's been solidified into place. So it's just like having a petri dish on a plastic stick. One side has media that will grow just about every bacteria. It's TSA, the same general plates that you used in lab. The second side contains a media called violet red Biol agar, which is selective and differential, and only coliform bacteria will grow on it. That's pretty clever. 2 different sides gives you an internal control. Everything should grow on the total aerobic bacteria side. So if there's bacteria present, we should see colonies. If there's nothing growing on the violet red Biol agar side, that tells us that whatever is present is not a fecal coliform. So here's our white side. This is made up of TSA, and it also has a chemical called TTC, which is a dye that turns red in the presence of bacteria, just so that it's a little bit easier to look at. So all bacteria is going to grow. An environmental water sample taken from somewhere without fecal contamination is all but assured to have bacteria growing on it. As I said before, the red side is selective and it will only allow coliform bacteria to grow. So in one simple test, by dipping this paddle into water, incubating it at 37 degrees overnight, we should see colonies. This will tell us if bacteria is present, and the violet red side will tell us whether coliforms are present. Now I said that these are semi-quantitative because they're not exact and they cannot give us an exact number of bacteria in CFUs per milliliter in the water. That's because all of the bacteria in a water sample don't actually come in contact to the paddle when we dip it in the water. The procedure for making one of these work is that you would open up the paddle tester, dip it in a water sample for 20 seconds or 30 seconds, shake off the water, and then whatever bacteria happen to settle on the paddle will grow into colonies. And then we just compare the paddle to a little scale that's present in the box that's been calibrated with lots and lots of repetitive testing. And here's where the semi-quantitative nature comes in. You just look at your paddle and you compare it to a number of the pictures that there are in the book. So if there's only 3 or 4 colonies, that means that the water sample likely had somewhere in the range of 100 to 500 CFUs per milliliter. Maybe 15, 16, 20 colonies sort of peppered around the paddle is representative of 10 to the third colonies. And then the denser the colonies get and the more that are present, that increases the estimate by an order of magnitude. So if I was to compare this paddle with colonies on the TSA side, we can see that they grew red because of the TTT indicator. And I would say that this paddle is more representative of the paddle that has water with 10,000 CFUs per milliliter than it is to 1,000. That's a pretty good estimate. I can say that there's 10,000 CFUs per milliliter approximately within an order of magnitude, and that's quite a few. You would do the same thing with the opposite side of the paddle to look at the estimate for total coliforms. So to finish up our completed tests, we would have had colonies that grew on a plate that we could gram stain. That's number 2. We have results from a phenol red lactose broth test. Now we can enumerate by diluting our water sample using a 1 to 10 dilution series, plating things on TSA plates so that we'll be able to get an accurate count, and also using paddle testers to see what the estimated bacterial load is semi-quantitatively. And then you'll remember that in our completed tests, we need to re-verify that bacteria growing in the EMB plates turn green and look like E. coli. So we'll just plate some of one of our dilutions onto an EMB plate and look for the presence of dark purple or greenish colonies. All of those bits of data together complete our tests and confirms that there is fecal contamination in our water sample of interest. So just to reiterate, a completed test is one that we are able to show that gram-negative bacteria are present, and we verify that with a gram stain, and that the bacteria are rod-shaped, which indicates that we have a coliform bacteria. That the bacteria is both gas and acid producing on phenol red lactose broth tubes, and that we are able to get an accurate bacterial count either through the dilution series or with our commercial paddles. And that's enough to confirm the presence of E. coli and therefore fecal contamination in our water sample. So where does that leave us? Well, make sure that you come back after 24 hours to take pictures of all of the results that you've collected this week in lab. And then you'll have a couple weeks to write up your results. Make sure that you take enough pictures so that you've got some things to edit. Make sure that they look really nice. Crop and zoom your microscope pictures. Make sure that your images show exactly what it is that you're trying to tell your audience during a presentation. And then watch out for one final video from me the week after Thanksgiving, in which I'll tell you everything that you need to know about your final project, all of your final due dates, and your final exam.